So Nora graduated from BU in 2015.
As an undergrad.
As an undergrad, yeah. And then she flew to the other side of the country to Stanford to do her PhD that she earned in last year, right? Yeah. And since then she joined Scale Bio as an application scientist since July. Pretty new. Yeah. I've been told that she does enjoy a lot and appreciate troubleshooting and helping people and scientists to perform their experiment to get their results. And I'll let the floor to you.
Thank you very much. I'll turn my mic on. Thank you for that lovely introduction, Nabil. Just checking here that you can all hear me okay. It's not too loud or anything. Just go like this if you want me to quiet down. So as you know, my name is Nora. I'm here with Scale Biosciences, and I'm really excited to show you all how we're unlocking single cell workflows with combinatorial indexing and all about single cell genomics at scale. So of course, I want to start off explaining some important applications of single cell sequencing. As you know, there's a huge range across biomedical and pharmaceutical research and that are really in need of high throughput sequencing. And some of these might include cell atlasing, which of course is cataloging cells based on criteria such as cell type, state in the cycle, location in the body, different transitions it undergoes, as well as lineage. Longitudinal studies, which of course goes hand in hand with large patient cohort studies. So for example, do you characterize chemotherapy resistance processes? We would collect tissues before and after chemotherapy and then analyze transcriptomic profiles at the single cell level to obtain that resolution. For CRISPR screens, which of course is everybody's sort of favorite needle in the haystack method of gaining an understanding of a small number of really important genes that contribute to different phenotypes of interest with knock-ins or knockouts, as well as rare cell profiling, which is really obvious here why single cell profiling is important, of course. So, for example, there are only maybe a few compared to the many other kinds of cells circulating in the bloodstream of a patient with solid tumors. And of course, in biomarker in relation to drug discovery, these can show associations with characteristic responses to any kind of therapeutic intervention. And so what I really want to underscore here is that single cell analysis methods provide this information that is generally masked by bulk methods. This intrinsic heterogeneity of kind of averaging across tissues that are used in bulk methods, you're really losing that resolution that you need at the single cell level in order to gain the insights that we're most interested in. So what goes hand in hand with this is this graph, which has been modified from Nature Protocols publication in 2018, really speaks to that, where we can see that the number of single cells used in studies are quickly increasing, and assays that reveal single cell transcriptomic profiles with greater resolution are in increasing demand. So this scaling upwards is part of what really inspired our founders, who I'm grateful to mention and be working as part of their company here. So there's Frank Steemers, Gary Nolan, Jay Shanduri, as well as Cole Trapnell. Scale Biosciences has released kits within the last year across a wide range of fields, the most recent one being single cell methylome sequencing. Of course, that's at the DNA level. And what I'm going to describe to you now is some traditional capture systems that people have used for single cell sequencing and how they're different from our combinatorial indexing. So traditional capture methods, as you may well be familiar, typically rely on microfluidics. And what these do is they compartmentalize droplets containing a single cell, some type of lysis buffer, and typically a microbead, which is covered with a barcoded primer, as seen here. However, one of the issues with these capture methods is that if two cells are captured within the same microfluidic droplet, you end up having cells that are barcoded with the same indices. And what happens in this case is that they can't be distinguished during sequencing, therefore your reads are not usable. And so you end up throwing out a lot of data, which is of course what you want to avoid. You're trying to get a better resolution here. And so even though these physical capture methods are really popular, combinatorial indexing, which is part of what we're introducing, is helping to get that resolution in a different way. And so how we do this is we actually put the barcodes in the plates of the cell. So there's no physical droplet compartmentalizing your indices. And instead, you have multiple levels of barcodes across several plates. So this first level shows you that there are 96 unique barcodes across the first plate. This second one shows you that there are 384 unique barcodes across a second plate. We all love pipetting 384 well plates, I know that. But it is really wonderful, the multiplexing capabilities that are unlocked through this. And finally, and I'll dive into a little bit deeper how this is done and show you the library structure. There is a third level of barcoding across this, another 96 well plate. And you actually end up with over 3.5 million unique combinatorial barcodes. And so what I'm going to describe to you now here is our really approachable workflow. I didn't mean to go past this slide here of our single cell RNA sequencing kit. So this workflow is really approachable. It truly only takes a couple of days. I've done it myself, so I can absolutely speak to that. Cell fixation only takes a couple of hours. Next is that plate-based workflow, which I described. We also employ smart plastics, which I'll describe in a bit more detail next. And we also have a full bioinformatics pipeline, which has an analysis-ready output for people to use. So if you're not a coding expert, neither am I, it's still pretty easy to understand the resolution of your data and how to use it for analysis downstream. So just to dive a little bit deeper into this barcoding section, we actually have validated our fixation. You know, things can happen in the lab, right? Other things come up. Maybe you realized your grant deadline is sooner than you thought, especially the internal one. And your long-term sample storage, which you might need, you know, oh, I actually can't get to this for a while, has been validated in the -80 for up to 6 months. So you can fix your samples, which takes a couple of hours, and then put it away for 6 months and begin again with absolutely nothing to hinder your results later on by starting the workflow. So to describe these levels of barcoding, I want to show you that across these three splits, there are three different kinds of reactions. So the first is reverse transcription, of course. You're starting with your RNA. You're going to create your cDNA from there. And I want to also mention that there's a lot of flexibility in this workflow. So it's usable with our Scale Bio custom RT primers. We actually have a protocol design which shows you how both to design your primers and to pilot them so that you're able to spike these in for further amplification. So for example, other targets of interest that might not be captured in the normal transcriptomic workflow, for example, if your sequences of interest are not polyadenylated, or perhaps they're too far from the 3' end of your mRNA to be captured by this traditional 3' single cell RNA library prep. We also have this option here at the end because we found that many users have a lot of cells left over after this ligation barcoding step, in which you can recover, typically you would recover through the normal workflow 125,000 cells, but you can actually recover up to 500,000 cells. This has been well validated. You can add on an optional 3 plates, 1 to 3 plates, in order to increase your output and basically bang for your buck. So you're not doing experiment after experiment, and you're conserving those precious samples. So I mentioned before that we have these smart plastics. And what I want to show you here are these really cool, I thought they were really fun to use, pooling funnels, and they facilitate your workflow by allowing a place to pool your cells after each round of indexing, as I showed you across those 3 plates. You pool them here, and they're in this specially designed funnel, which is super easy to centrifuge, and it really decreases your cell loss, which we know is a huge part of some of the issues with single cell sequencing workflows in general. And as promised, I wanted to show you a little bit deeper the library structure. So what you've got here, as I showed you before, are how we generate over 3.5 million unique barcodes. So you start off with your 96 barcodes here, and you have an indexed reverse transcription, I want to point out that like other well-known sequencing platforms or library prep platforms, we also include unique molecular identifiers in each of these wells. So not only are you getting the 96 unique barcodes, but there are thousands of UMIs, unique molecular identifiers, within each well to identify each original transcript. And this really helps you down the line to show that you actually have more than one transcript of the same kind rather than just a repeat. So it allows you that greater resolution. You're probably well familiar with this. Next, for your index ligation and your second strand synthesis, you're turning your cDNA into double-stranded DNA here, and you're adding on, in addition to that barcode that's already there. The mic fell. Give me a sec. Thank you for your patience. So in addition to that barcode, you're adding your ligation barcode as well as your partial read one. And finally, after that part, you are now moving on to your tagmentation and your indexed PCR. And this is where you're creating the full library molecule that will go into sequencing. So at the end, what you'll have here is your RT barcode, your ligation barcode, your full read one and read two sequencing primer binding sites, as well as those flow cell complementary oligos, P5 and P7.
I can ask a question. With the tagmentations, how is that controlled so that you only put the tag at the end and it doesn't like cut in the middle of the molecule?
So that is you're talking about here?
The last oligo said tagmentation.
Yeah. So as far as I understand, that's done because there is an overhang, and that should be recognized only at one end, and so that's why you're only going to get your break there. So it's not gonna break in the center of your molecule, and I believe part of that has to do with the fact that this part as well is polyadenylated. But I'm happy to follow up in further detail if you'd like. So now I'm going to describe to you a bit about our data analysis pipeline and how we provide these analysis-ready outputs. So as you're probably familiar with, Illumina generates these binary base call BCL files that are produced and turned into FASTQ files. That's done using the well-known BCL convert software. And then for each cluster that passes filtering, right, one sequence, for each cluster that passes filtering, one sequence is written to the corresponding samples read1 and read2 files, and then you get a pair of files for each set of this paired end sequencing. And then for barcode parsing, ScaleBio, we have our own executable, in which these 3 single cell barcodes are extracted from the reads, and then finally, sample reports are generated, both on a bulk level and at the individual sample level. And these summary reports include metrics for each sample, such as mapping metrics, cell counts, and sensitivity metrics, as well as a distribution of RT barcodes for each specific sample. The output file is in both a very readable HTML document that has graphs and tables, and these values are also available and generated through CSV files with metrics and text format. I also wanted to let you know that if you want to explore any more of this, all of this in our pipeline is open source on GitHub, and we will be really happy to direct you there if you would like. And so now I just want to dive a little bit deeper into this multiplet rate, which I had mentioned before was an issue with traditional capture methods. So doublets or multiplets, these cells that are barcoded with the same sequence or transcript should appear to originate from a single cell, and that's why they're problematic. But we, of course, use barnyard experiments, which are the typical validation of single cell technology, proceeds with a mixture of two species, typically human and mouse cells, such as in this case. In this case, a subset of wells, 10 wells, were taken through sequencing, and those cells that whose transcripts are matching to both mouse and human, mouse and human references are, of course, not truly single-cell data, and we can consider those as multiplets, which are pretty easy to see here. What's really nice is that we have quite a low multiplet level compared to some other platforms that go higher than this. In this case, it was only about 3.4%. And I also want to highlight that in the background, there's only about 2.83% of those cells, which were kind of whose reads can be contributed to ambient DNA and not usable DNA within your cell. And now I'm pretty excited to show you some real customer data from macaque brains. These are brain-derived nuclei and cells, and they show a nice sensitivity and good integration with the reference data that's already existing. So as you might have guessed, they were loaded onto our workflow across these 3 levels of barcoding. And I want to point out that although we're pretty certain that you can get 125,000 cells recovered here, they actually were able to recover more than that at 188,000 cells. This group is, of course, at UCLA. And what I want to show you here as well is that it was fairly consistent across samples in terms of what the reads looked like and in terms of metrics. So something important to notice, as I mentioned before, you're not having all these wasted reads like going to background, whereas over 88% in sample 2, your percent of reads that are actually within cells is really, really high as opposed to going to backgrounds. And to convince you further, as you know, the brain is composed of highly heterogeneous tissues. And what I'm showing here is that we're able to pick up those typical clusters. So this assay can really push through a lot of samples. And not only is there lower ambient DNA in our prep, but if you were to sequence deeper, there's certainly more data that you can find, given that the saturation is not yet fully exhausted, right? There's still upwards mobility there at 0.32 saturation. And at the same time, you're still getting the resolution of these clusters. I also want to mention that we have a webinar that goes over this data. So if you would like to take a deeper dive, we'd be really happy to send a link out. Next, I'm excited to show you a little bit more customer data. I think this is really interesting because I've talked a lot about the existence of other platforms before, but now I can actually show you side by side what an unbiased experiment from an outside group actually looks like, comparing these sequencing platforms between one another. And so this was conducted by the Children's Hospital of Philadelphia, and it was presented at an ASHG poster very recently, which compares these platforms. So just to give you a little bit of background here, the single-cell RNA libraries were generated from 4 PBMC donor samples. These were prepared using methods according to each respective platform's protocols. As you know, Scale Bio's workflow time is 2 days total, and then these other platforms here have a varied amount of time that their workflows last. I need to mention that the PBMCs used here were particularly challenging. They were from patients, and they were lower quality than the commercially available stocks that people are often using in lab. And the resulting libraries were sequenced on a NovaSeq. And so what I'm just showing you here is those differences in storage time. Across the 4 platforms, we obviously have the most flexible storage time. In this case, they started processing these other samples pretty fresh from day 1, whereas they began processing ours after they were in the -80 for a couple of months. And just highlighting here that these four circles are representing the 4 human PBMC donors. So now I'm going to show you a little bit about the cells called by the respective platforms. You'll notice here that our cell calling is is higher than some of the other samples, spare really one exception. However, I think what's most notable and honestly most important, because we know, the highest number doesn't necessarily always mean the best. Consistency, although we do have the higher numbers overall, is really important. So what you can see is that even in this center platform, which uses droplet-based sequencing, and on the right here, this platform uses plate-based sequencing, which is a bit similar to what we have. We're getting a lot more consistency and number in our samples. So across these 4 different sample types, this shows that there's reproducibility among your data, and it's not kind of a wash and a guess over what you're actually going to get. So I think that's really important to point out, because obviously we all know that we're in a reproducibility crisis here. It would be nice to be able to actually reproduce your data, certainly among a sample set, which is what we're hoping for. I also just want to show you this multi-metric analysis across platforms. You'll notice that there were originally 4 platforms that that the Children's Hospital of Philadelphia were comparing, but for some reason that other platform was not actually able to generate any libraries. So that's why they're not included here. I just want to make that clear. But we demonstrate, really high performance, this consistency, as I mentioned. I also want to show you that the reads within cells, which again tells you that your sequences, your time, and your money are going to your biology and what you're trying to figure out, as opposed to, just kind of wasted ambient background. And so given that these other platforms appear to be showing some kind of batch effect by each run, this really presents challenges, for example, for longitudinal studies. And just to dive a little bit more into how we know and how we can see that there's a good marker for the fact that these other platforms have a greater background reads that they're getting, I'm going to show you a bit about the mitochondrial percentages within the reads on this violin plot. So in particular, I'm highlighting our performance next to a droplet-based platform, and we seem to have a much greater sequencing efficiency. If you're wondering why, we're not completely certain, but one of the hypotheses for why we're able to get fewer mitochondrial reads is that our kit does include a series of wash steps, and as the cells are exposed and they're not just contained in these droplets that can't necessarily get their ambient bits out. So now with the wash steps, you're probably less likely to capture backgrounds such as mitochondrial RNA, and you're more likely to capture this in a droplet-based format. And just to give you a bit of an overview here, the success of our PBMC library generation showed that although across the samples, most of the platforms except for one were able to generate libraries, the library quality overall did differ, whereas Scale Bio had the highest transcriptomic mapping rate. The fraction of reads in cells was a bit lower on the droplet-based platform, but it still was substantially higher and consistent when compared to this plate-based platform. And this, of course, translates to fewer wasted reads. The size of the library really shows that we're not yielding any kind of poor quality data. The sizes were as expected. And our cell recovery, as I mentioned, was the most consistent. And proportionally, of course, mitochondrial content was dramatically lower compared to this very popular droplet-based platform. So, as I mentioned before, yeah.
Before going forward, can you comment on genes detected, and was there a lot of overlap between the platform or not?
In terms of which genes were detected,
Yeah.
I'm actually not sure of that information, but I am happy to follow up.
Yeah, like how did the UMAP plots compare between all the 3 different platforms?
I don't have them here, but
You said you supposed to.
yeah.
Yeah, genome lab was pretty good on 10x scale. I can send you the poster.
Okay.
Great question. Okay, so I also want to mention here, which is pretty exciting, our fairly new CRISPR screening kit, which we've released. This actually works in tandem with our single-cell RNA workflow. I might not need to remind people, but I'll go very quickly over some typical CRISPR screening methods here. So, as you might know, in an arrayed screen, your guide RNA is introduced to your CRISPR-Cas expressing cells. Each well typically contains a unique gene target. Cells are perturbed in separate wells with about one target per well. And these readouts are done by imaging, proteomics, or metabolomics. This is an advantage, of course, because you don't actually have to sequence and the quantification is not required. But of course, it ends up being pretty labor intensive and costly. And given that there's only one target per well, it's somewhat limited in scale. Some of the applications, as you may know already for these, include the study validation and confirmation after larger pooled screens, which I'll describe next. So in these larger pooled screens, of course, guide RNAs are introduced to CRISPR-Cas9 expressing cells through typically a viral lentiviral transduction. Guide expression facilitates these target gene changes along with some kind of a challenge, for example, a drug treatment, and your sequence and analyses identify and enrich your depleted genes, enriched or depleted genes. Some of the benefits, of course, that these are lower costs and labor than arrayed screens, and they're considered high throughput. However, they might be a bit less accurate, and they rely on readouts that can impact cell viability. This is great for high throughput screening, of course, if that's what you desire, as well as genome-wide screening, discovery studies, and perturbations across wide pools of cells. Unsurprisingly, you're going to hear me explain why single-cell CRISPR screens provide the greatest resolution here. So in this case, as guide RNAs are inducing perturbations and they're also serving as genetic barcodes, you can obtain, pretty precise quantitative representation of guide RNAs by sequencing their amplicon libraries overall, and it really accommodates massive guide throughput. The applications really go across the board for high throughput screening, genome-wide screening, as well as study validation. And just to show you a bit here, you can see that overall, you're really getting the most scale and insight out of your single-cell CRISPR screens as opposed to, say, pooled and arrayed screens. And so now I'm going to briefly take you through our workflow for these single-cell CRISPR screens and how they work in tandem with our single-cell RNA sequencing kit. So, of course, you will have the freedom to be designing your CRISPR library the way that you want. You would typically assemble these cells using a lentivirus to combine your Cas9 with your guide RNAs of desire. Then you would transduce these into your cells, select the cells that were successfully transduced, and then you would take them through the single cell RNA workflow in pretty much the same way that I had described earlier. There is another PCR included, which will further amplify your guide RNAs. And then you would take it through sequencing, and of course, Scale Bio's own CRISPR bioinformatics pipeline, which will again give you analysis-ready outputs. Just a little bit more depth here with the text workflow after transducing your cells, which, by the way, we are using a crop-seq vector, which you may be familiar with for gene knockout, knock-in. You would pull your cells and then distribute them across that first RT plate, UMIs, of course, included. After this, you could use both the poly-A, the typical primers that you're using for your RT plate, which is specific to the Cas9 studies, or you can use a custom primer. As mentioned, that flexibility that was available in the single-cell RNA-seq workflow is also available here, which is really important. As I mentioned, sometimes you want to be amplifying either other targets alongside your guide RNAs, or your guide RNAs might be located in a place where they're not getting as amplified as you'd like them to be. So I also want to mention that you see Cas9 is here, but this is not just limited to Cas9 systems. You can use other Cas systems with this workflow. Afterwards, you would go on to the rest to sequence both your transcriptomic mRNA as well as your gRNAs. And then you actually take an aliquot of your RNA library pool after this, after that final barcoding step. And what you're going to do is do that subsequent PCR, which is specifically to enrich your guide RNAs, and then you would be enriching the rest of your library in the more standard way separately. And as I mentioned, this can be helpful to amplify signal from targets that aren't polyadenylated or too far from the 3' end of your mRNA to be captured. Finally, after that, you'll be able to identify your guide RNAs of interest at a really great resolution. So this is really similar to the figure I presented before. I won't go through the whole thing in depth, but I do want to mention that the fixation here is really useful for single cell CRISPR screening in the sense that there's some more complex screen designs and flexibility regarding the conditions and that you're creating. And with this extended sample stability, it really gives you flexibility in your workflow. Finally, as I mentioned, that RT barcode, the flexibility that is allowed there using the custom RT primers, which we have that protocol for you, which shows you how to pilot and design those primers, is really useful for spike-ins. And of course, I also want to mention that the extended throughput kit, which enables further recovery beyond 125,000 cells, is also available as an option here. As I mentioned, your additional PCR reaction for guide amplification. And finally, if opted to, you will also have a sequencing output of up to 500,000 cells, many of which will hopefully be your guide RNAs. And just to give you a bit of an overview of how this might compare to other single-cell crop systems, which you may be familiar with, they typically just use the poly-A primer. In a comparison that we did with human cells, we saw that 42% of reads, which were containing a real guide, was all you really got out of the cells, just 42% of them, compared to 93% of reads, which contained reads for a real guide RNA. Finally, in terms of cells that could actually be associated with a guide RNA, only 67% of cells had a guide with the traditional CropSeq protocol utilizing the poly-A primer, and over 90% of cells in Scale Bio's case were recovered with a guide. So as you can see, we have an increased sensitivity compared to those traditional methods, and a decrease in sequencing cost, because by having these pure libraries, you don't have to sequence quite as deep because you have this nice pure library. And I also want to show you, you might be thinking, well, what do you consider like a cell with a guide, right? What's your threshold? What I can show you here is that our system actually allows for quite stringent thresholds compared to more traditional CropSeq systems. So what you can see is, for example, in this case, we would typically call a true cell with a guide would have 3 molecular identifiers. That's where that orange dotted line that's the typical threshold we were using. In that case, we've got over 70% of cells recovered with a guide. However, if you want to maybe be more stringent, say you're not sure if those data are real and you want to make sure that there's actually a lot more recovered with a guide, you might move that threshold all the way up to, say, 30 UMIs or 25, and you're getting still about 30% of cells that are recovered with a guide. However, if you're using a traditional CropSeq system, using just poly-A capture, what you're getting at, say, a cutoff of 30 is essentially 0% of cells with guide. You just can't be as stringent, and therefore you just can't be quite as certain about your data. So I just wanted to speak to, I think, the strength of this system, and how it compares to a more traditional one. And so now I'm really, really excited to tell you a bit more about another kit we have. We're turning in a different direction now, away from RNA to DNA, of course, which is our single cell methylation kit. As you probably already know, if you're checking out the literature, there's really a ramping interest in profiling methylomes at the single cell resolution, especially. There are, as you know, approximately 30 million CG methylation sites in the human genome, and they have been found to be involved in many different kinds of regulatory processes, from transcriptional regulation to cell differentiation, X chromosome inactivation, as well as association with diseases like rheumatoid arthritis and neurological diseases, as well as cancer. So, of course, methylation at other sites is considered important, even though it's not quite as common in the genome. CH methylation at cytosines, followed by any other base that's not a guanine. And what I'm going to explain here is a little bit about the applications of single cell methylation. So, of course, in oncology, there's the option to look at methylation data from liquid biopsies. Aberrant methylation signatures in disease research are likely to offer novel insights across research as well as translational use cases. In precision medicine, these signatures are emerging as important biomarkers for biomarker development and patient screening. In longevity research, many people have shown that epigenetic factors are associated with age, especially methylation controlling the cell and gene regulation. And there's a large body of work showing that methylation signatures are associated with these changes in age. But single cell resolution would be really useful, of course, to elucidate these data that are already existing. In neuroscience, cognition research, there's of course the exploration into how methylation patterns are driving long-term and short-term memory. In neurodegenerative diseases, epigenetic factors are being explored for their contribution in dysregulated cell function and in manifesting in diseases like Alzheimer's and dementia. I'm sure you're also familiar with the applications of methylation across stem cell research and therapy, as well as developmental bio generally, where epigenetic changes may regulate cell fate, and this different programming and modifications within these different subtypes are being explored in order to manipulate cell lineage. And of course, in development, development may be governed by this very complex regulation and coordination of stimuli, which can be signaled by methylation across many stages. And so what I want to highlight here is just that cell type specific data around these kind of methylomic alterations would really provide a lot of great insight into this huge variety of fields and applications. And so what I'm going to show you here is how Scale Bio actually manages to use these existing methods that we can rely on, for example, bisulfite conversion. That's what we really employ to detect our methylation signatures, along with the combinatorial indexing, which uses barcoding that across a plate-based workflow, which will end up giving you methylome signatures at a single cell resolution. So a quick overview of this kit. It is a completely kitted solution. We've got a lot of documentation and product support available. This is also fixation enabled. It's a bit more limited sample storage time than our other kits, but part of the reason for that is that this simply hasn't been around as long, and so we're actually still validating that sort of storage time. The workflow is fairly streamlined. I want to mention that we do have a really talented field application scientist team. So for any of these workflows that I have mentioned to you, for your first run through of any of these kits, we will send out somebody to go through the workflow with you on site. So I also want to mention that we can detect, heterogeneity of methylome signatures across samples. So we're able to detect both CG and CH methylation. And finally, as mentioned with our other kits, we do have a complete analysis pipeline, of course, unique to methylation. It's different than our RNA pipelines. And these are run with, good QC summary outputs with some familiar metrics like you would like to see, of course. Some of these familiar QC metrics, as well as, which are adapted for methylation, as well as a matrix output, which shows you your cell ID and then methylation signatures across it. Some of these can be configured across genome binning, gene bodies, and other methods of analyzing the methylome data for any type of analysis that you would like to do. So you're probably wondering, okay, great, they have a methylation kit. We've never heard of this before, single cell methylation. Part of the reason for that is that we are the first ones to release this kind of thing. But I am also able to tell you that there are many different sample types that we've actually validated this on. The list is only growing longer, but these are what we have validated so far. Cell lines, PBMCs, human brain cells prepared both frozen and OCD prepared, as well as human kidney samples, mouse kidneys, and human testes. I do want to mention that despite the fact that there's quite a variety in cell types that are validated, the minimum requirement for the number of nuclei actually remains the same across these different cell types, and that's been well validated.
What's OCT?
OCT, I forgot what it stands for, but it's like a if you fix your samples in it.
Is it fixed?
Sorry, fix isn't the right word. It's like a
Fixed.
frozen in a specific compound.
Just
It's cryo, yeah. And so just a quick overview of the workflow. After doing your fixation, which is actually PFA based, then you do a nucleosome depletion. Of course, your DNA is very tightly packaged, so you're going to need to loosen that up a bit, deplete those nucleosomes in order to expose the regions for tagmentation. This is where your first level of indexing happens. I will show you the library structure on some following slides. After this, you would be doing a nuclei sort and a distribution to a 96-well plate. This is done using FACS sorting. Next is when the bisulfite conversion comes in which your non-methylated cytosines are turned into uracils during this conversion, and those that are methylated remain cytosines, so that when you're looking at your analysis, everything that's a cytosine is methylated. Finally, you generate the rest of the library through a second level of indexing, and you attach the necessary sequencing adapters, and I'll show you again in the library structure. And finally, you can sequence up to 2 to 8 samples. This kit actually comes in 2 sizes, both a small and a large, and then you can do your data analysis from there using our pipeline. So just to give you a quick overview of the library structure, you start off with your tagmentation barcoding. Here's your double-stranded methylated DNA. In this part, you're adding your tagmentation barcode as well as your TrueSeq read 2 sequencing primer binding site to your 5' ends. Next is bisulfite conversion, in which you are you're actually cutting off. You can see that 3' label, but that will get restored later, where you can see you've got your DNA now split, and you are converting your non-methylated cytosines into uracils. Next, you would perform a splint ligation step. And this is when you're adding your methylation adapter primers. And during the splint ligation, you'll be attaching a partial read 1 sequencing primer binding site to your 3' ends. And of course, your barcodes are maintained. And finally, in your indexed PCR, at the end of this, you will have your complete library structure, which includes the I7 and I5 indexing barcodes, and I'll explain the amount of those that we provide in a minute, as well as your flow cell binding sites, your P5 and P7 oligos, and again, your read 1 and read 2 primer binding sites are also included there in this double-stranded product. So just to show you a bit of a deeper dive about this combinatorial indexing for this workflow, we provide 12 different I7 indices and 8 I5 indices, which, as you can see, are distributed across the plates as followed, which is nice because, of course, you can do that using a multi-channel. When the multi-channels behave, of course, they are a really, really useful way to save some time. And though we have 96 unique combinations here, what I'm going to show you now is that given that you're doing your tagmentation across a few different plates, across 3 plates total, you will have 288 tagmentation barcodes combined with these 96. And that's how you get to over 27,000 combinatorial barcodes. And so this is what your final library is going to look like, as I showed you before. I'm just going to dive briefly now into the analysis workflow. We will be on GitHub here soon with a very solid example data set. We'll be happy to send this over once it's ready. And again, this pipeline is open source. So that will be available for you to check out and explore on your own. So for the analysis workflow, we of course have our run QC, which includes fast Q generation, read QC, and adapter trimming, your sample QC, which includes your sample barcode demultiplexing and collapsing, as well as genome alignment and read deduplication, as well as cell thresholding. As I discussed before, you'll be extracting those methylation signatures from your data, and they will also be generated in a matrix, which is great for analysis, as well as final clustering and differentially methylated region, or DMR calling. And as I mentioned here, looking forward to getting a finalized data set there for you, but I do have a preliminary data set to share with you. Of course, I wouldn't share this without data. So just a quick preliminary summary. The performance of this workflow appeared to be quite robust across sample types and species, across human and mouse tissue. As I mentioned, this is the overview of the different tissues that we used before. I want to show you, in real data that the workflow is in fact applicable to different sample types. The ratio of unique to total reads in both human and mouse species is going up in the upward trend, as you of course would like to see. With as you get greater total reads, you're also increasing the number of unique reads you're getting. I'm going to jump to this on the right here, which is across your different tissue types as well, so slightly better resolution here in terms of what we're actually looking at, not just species. You can see that across these very varied sample types, as your total reads are going up, so is your number of median unique reads. And finally, your ratio of CG methylation coverage over your unique reads is also going up. So as you're getting more unique reads, you're able to cover more CG sites. And so a quick summary of the data as promised. What you can see here is these were PBMC samples, human, again, across these 3 plates. That was the tagmentation setup. And what I'm showing you here, I want to point you first to these graphs down at the bottom. So we typically would expect these levels of both CG and CH methylation, with CH being less than 1%. And CG being greater than 70%, as typical in the human genome. Something I want to mention here is that if you're seeing like a really, really high level of CH methylation in a data set, that's bad, because that would denote an incomplete bisulfite conversion, where you're, instead of turning into uracils, right, your unmethylated cytosines would remain cytosines. So that would be false. But again, this indicates a complete bisulfite conversion, which is really good. Similarly, regarding the number of cytosines covered, for CG methylation, this is typical. The reason why we're seeing so many cytosines covered as expected for CH is, of course, because realistically, right, a C is just statistically more likely to be followed by an adenine, a thymine, or another cytosine. So that's why it's technically covered more, right? So we anticipate this higher coverage of the CH sites, but we only anticipate actually, less than 1% of these to actually be methylated. Something really important I want to highlight here as well is the fact that our benchmark for the number of CG sites is about 500,000, but we were able to achieve 700 sites covered of CG methylation sites. And this is just really important to show you the robustness of the workflow. Similarly, the number of reads that we expected was similar in both CG and CH methylations, with the number of unique reads in CH sites being, of course, less than the CG sites. And finally, on this knee plot here, what you can see is a really neat separation of our signal to background with this clean drop-off. We know what is real usable real and usable sequences through these unique counts versus the number of barcodes they had. And you can see that through the drop-off here. So just to summarize that kit right there, methylation in general, we have they're storable, transportable, and batchable samples here for our methylation workflow. It's also a streamlined workflow with varied cell output depending on whether or not you choose to use a small or large kit, with a cell throughput of 4,600 to 18,300 cells per run. We use this powerful chemistry, bisulfite conversion, that we already know and have been well established to work, as well as the fact that I really should mention that this is also target enrichment compatible. So if you're looking to amplify specific targets, you can certainly do that with this kit, which will overall reduce your sequencing costs and preserve your time and energy for your reads going to really usable data. And finally, you're able to multiplex up to 8 samples per run. And now that I've finished describing our platforms here, I'd be really happy to take any questions. I do want to mention that after the lunch break, I will be going into a bit about transitioning from academia to industry if people are interested in that. I've got a few slides set up there, but I'm happy to transition over and take some questions now.
Cost. How much does a scale kit compare to the other platforms in terms of cost?
For RNA?
Yeah, for typical single cell sequence, RNA sequence.
So our base kit for the 125,000 cells in nuclei is about 8,800. You can extend that to 500,000. It's a little variable after that. We do discounting for academic teams and first time.
So the starter kit is 8,800 for how many samples?
125,000 cells. So you can technically do 96 samples. We usually see 12 to 30 on a plate.
But you don't have 96 samples, let's say you only have 12.
You can do 12, yeah. So there's 96 unique
That's so-
indices. So you technically could do 96, or you could just do one sample in a full range. It just depends on how you load.
Because you can do like greater aliquots, right? Multiple aliquots of the same sample across your plate.
What I'm doing. It's what I'm doing, running 14, I think.
14 samples?
Yeah.
Okay. But they don't come in smaller sizes. It's 8,800 is the base.
That's
kit.
the base, that's the list price.
Okay.
And again, we do discounted practice.
Okay.
How about methylation kit?
Methylation is a little different, especially when you take into account the sequencing needed per cell. I'd say it's around $2 per cell, and then another $1 per sequencing. So the smaller kits are around $10.
For the methylation case, I have a question. How can you know which cell is coming from? Because you're looking just at DNA, right?
Yes, DNA.
So do you have UMAP to know that, let's say you have tissue where you have
Yeah.
I do have
different
I do have-
subtypes? Do you have
There's a
UMAP?
few different ways you can do it. So
Slides.
you can do like 50 kb binning.
Here.
Just get
Oh, okay.
So there's some
So-
PBMC. I
So
also
it's
I have another
full genome
one.
So you do RNA and
So actually, you're just doing
This is just methylation.
just yeah
Multiomes next year.
yeah. It's just DNA with this one. Yeah, but I understand it's like jumping over. Yeah, but like here, right? You can see your CG methylation percentages, and then in that same map, you can also break that down by cell type on the left.
Don't you have to do both at the same time? You
How-
need the RNA-seq to tell you what cell type is.
So yeah, this is an approximation. It does map pretty well to PBMCs with RNA, single cell RNA workflow. It's just not as built out in terms of that flow. The data sets aren't as built out yet.
yeah,
We
because
do have a few teams that will be running it concurrently.
Yeah, we have. I think this
so
was,
if you
it wasn't done. Yeah, it was done simultaneously, but not within that same kit. Is what I'm trying to say.
Not the same team.
Right. So you're combining the RNA and the methylation data.
Yeah, you always have to do both, right?
so you have to run the RNA and then you combine. No.
Yeah.
You're saying
you run
this-
methylome, you run RNA. They're not currently combined yet. That'll be
No.
next year.
No, I mean
You
but
run them concurrently.
you can't. So you cannot really say that, let's say, in this group, without this data, just with the methylome data, say that this is T cell.
It's tough.
Yeah. Okay. So it's really
You don't have
great.
RNA data
Yeah.
directly correlated.
Yeah. So you need to put it. It'll be really powerful when you can combine both.
Yeah.
Because
Next year. They've
Yeah.
only been out for a year, so give us
Yeah.
some time.
Give us some time. Yeah, we're working releasing kits for a year. So.
What's the conceptual advance in the scales workflow that maximizes the reproducibility versus parse? Is it that funnel collection thing, or what's the technological difference?
We're not
Yeah,
sure.
we're not really
Like,
sure.
We can't comment on their tech.
Yeah.
The funnel definitely helps with cell recovery rates.
yeah.
And then you do poly A, and I think they're random expert with poly A. But yeah, can't really comment on their technology.
Yeah, don't know about the technology.
Okay.
Yeah, vague answer, but yeah.
Yeah.
We have to rely on just user data sets to show results. We're not totally sure.
With your, with the pipeline that you have for the analysis of the methylation, can you, within that pipeline, then go in and look at, say, once you've identified one of those cell types, the actual base level of the methylation?
By cell, like
Yeah, so-
yes,
so you-
yes, you can. This isn't telling me. But I do know that the matrix that's generated is the methylation signatures, so like methylated, right, your cytosines per each cell, like cell ID down to the single cell level. That I'm aware of, but I haven't seen or used the pipeline myself. Unfortunately, not a bioinformatician, but I do hope to familiarize myself with that greater. And as I mentioned, we'll